Hello students. So today we'll be studying the placenta and umbilical cord. So we'll be covering the competency, describe formation, functions and fate of chorion, amnion, yolk sac, allantois and decidua. Of the yolk sac we have studied, amnion we have already studied. Then describe the formation and structure of umbilical cord. Then you should be able to describe the formation of placenta, its physiological functions, feto-maternal circulation, the placental barriers. You should be able to classify different types of placenta. Then you should be able to describe the embryological basis of twinning in monozygotic and dizygotic twins. Then you should be able to describe the role of placental hormones in uterine growth and parturition. Then you should be able to explain the embryological basis of estimation of fetal age, which we have already done earlier, that is by counting the somites. And then you should be able to describe the various types of umbilical cord attachments. So firstly, what is placenta? You get a long question on describe the placenta. So placenta is an organ that performs exchange of nutrients and gases between mother and fetus. It is feto-maternal in origin, that is both the components are forming placenta, the fetal as well as the maternal component, and it is a characteristic feature of eutherian mammals which are feeding their young ones. Then according to its shape, it is a discoid and it is a choriodeciduate organ since it is shedded off, so it is a choriodeciduate organ. Then it has two components, that is a maternal component which is derived from so we were studying about the placental components. So with the maternal component is derived from uterine endometrium, so it is known as decidua, and the fetal component develops from the trophoblast and extra embryonic mesoderm, so it forms the chorion. So placenta with fetal membrane is also expelled out of the uterus 30 minutes after the childbirth. So that is why it is choriodeciduate organ, because the fetal membrane as well as the maternal component both are being expelled out. So what are the gross features? There are two surfaces over here. You can see according to the shape it is discoid, and there are two surfaces, the fetal and the maternal surface. The fetal surface is shiny, it is gray in color and translucent because of the presence of amnion, and it is covered by amnion, I have already told you, and it provides the attachment to the umbilical cord. This one is the maternal surface, which is divided into various lobes, which is known as cotyledons. So maternal surface is dark maroon and rough. It has a cobblestone appearance. It is divided into 15 to 30 polygonal lobules that are separated from each other by deep fissures. So these polygonal lobules, they are known as cotyledons. So fissures which are separating the cotyledons are occupied by placental septa. So you can see these are the placental septum dividing each lobule or cotyledons, and each cotyledon mostly contains a single stem villus along with its various branches. So this is the main umbilical cord. From here is the umbilical arteries branching. They are forming this different stem villus, and one cotyledon will have one stem villus with its branches. So it is the placenta is around 15 to 20 centimeters in diameter, and it is 3 centimeters thick, and its weight is about 1/6 of the weight of the baby, that is 500 to 600 grams. Now what is the structure of placenta? So placenta, it consists of a chorionic plate which is towards the fetal side, a stem villi, I already told you, then the intervillous space filled with the maternal blood. So this is a stem villi. These are the different stem villi, and between these two villi will be the intervillous space which will be filled with the maternal blood. Then there is a basal plate which is towards the maternal side. So placenta shows basal plate on the maternal side and the chorionic plate towards the fetal side. So firstly, the basal plate or the decidual plate. So the decidual plate is formed by stratum spongiosum of decidua basalis. Okay, so there are three layers: stratum basale, spongiosum, and compactum. Compactum may we have implantation going on. So the decidual plate is being formed by stratum spongiosum of decidua basalis. Then what else is there? There is cytotrophoblastic shell. We have already read that there were anchoring villi, and because it has to anchor the cytotrophoblastic layer, it will multiply, it will overgrow all the villi, and it will encroach the maternal endometrium, and it will form a cytotrophoblastic shell to enclose whole of the developing embryo. So a decidual plate is having stratum spongiosum of decidua basalis and cytotrophoblastic shell. Then there will be a syncytiotrophoblastic layer beneath it. Now, what are the components of the chorionic plate? So, the chorionic plate consists of extra embryonic mesoderm. So, this one is the chorionic plate, magnified extra embryonic mesoderm with the cytotrophoblast and syncytiotrophoblast. So, outermost is syncytiotrophoblast, then cytotrophoblast, and the core is of extra embryonic mesoderm. So, that forms the chorionic plate. So, in a gist, structure of placenta has two components, maternal component and a fetal component. Then the maternal component forms and includes endometrium. The stratum spongiosum will form the decidua, and the decidua is divided into three parts: decidocapsularis, parietalis, and basalis. Basalis is towards the maternal side of the growing embryo. Parietalis is covering whole of the uterus, and capsularis will cover whole of the embryo. We'll see this later on. And the fetal components are made up of trophoblast and extra embryonic mesoderm, which both will form the chorion. The chorion is also divided into two types: chorionic frondosum, which is at the embryonic pole, and the chorionic leaf, which is at the embryonic pole. Then the chorionic frondosum is separated by intervillous space, and it will give on branching, which will form chorionic villi. There will be three types of chorionic villi throughout the gestation period: the primary, secondary, and tertiary at the term pregnancy. Then their components, their branching pattern is made up of truncus coriae, ramus coriae, and the ramuli coriae, just like a tree. So whole of these two components will constitute the placenta. So now the decidua. The uterine endometrium during pregnancy is called decidua because it is being shed off. So decidua forms maternal part of the placenta, and the word deciduous means falling off or shedding in the Latin. So decidua sheds off during parturition or childbirth. There are three regions of the decidua as follows. Firstly, the decidua basalis or serotina, the part of the decidua that lies deep to the blastocyst. This was a blastocyst. The decidua basalis forms the placenta. Then is the decidua capsularis. We all know that extra embryonic mesoderm and the cytotrophoblastic shell, it was enclosing whole of the fetus. So the decidua capsularis or reflexa is the part of the decidua which will cover whole of the blastocyst. And finally, the decidua parietalis, later on the syncytial trophoblast will extend towards whole of the endometrium. So whole of the uterine cavity will be forming the decidua parietalis. Later on, as the embryo grows, this decidua capsularis will fuse with the decidua parietalis or vera. So decidua parietalis lines the uterine cavity. And capsule, from capsule you can remember as the capsule, it will enclose the embryo. Now, what is the decidual reaction? Whenever there is the implantation going on or embedding going on, there are certain cellular and vascular changes in the decidua during pregnancy, and that is known as decidual reaction. The endometrial cells, under the influence of the maternal progesterone, which is being secreted by the developing placenta, they get overladen by the glycogen and lipid deposits. So the cell size increases, and this provides nutrition to the growing embryo until and unless the uteroplacental circulation is established. So these are pale staining decidual cells. So a high progesterone level in maternal blood is responsible for this decidual reaction. Now, what happens in the junctional zone, that is the trophoblast and the junction between the chorion and the decidua. So, the trophoblast and the decidual cells, they will intermingle. So, this zone is characterized by decidual as well as syncytial giant cells, which are rich in amorphous extracellular material. Now, the development of placenta. So how it develops? So trophoblast, initially it was forming syncytial trophoblast. It will proliferate. It will penetrate the maternal endometrium. Okay. Then on complete invasion of blastocyst, decidua can be divided into decidua basalis, capsularis and parietalis. Okay. Later on, when the size of the fetus increases, this capsularis will fuse with decidua parietalis. Now, the trophoblastic layer differentiates into the outer syncytial trophoblast and inner cytotrophoblast. Now, then the uteroplacental circulation, how does it establish? Now, there was hypoblast cell layer. This is the hypoblast cell layer, which will multiply and it will form extra embryonic mesodermal layer. So this yellow one is the extra embryonic. This was the Usener's membrane. This only will give rise to extra embryonic mesodermal layer. Now these syncytiotrophoblasts, cytotrophoblast and extra embryonic mesoderm will form the chorion. Okay. Then the lacunar stage will appear. The lacunar spaces will start appear in the syncytiotrophoblasts, which we have already seen in second week of intrauterine life. So these lacunar spaces are more towards decidua basalis initially. Now the enlarging spaces, they will communicate with adjacent ones, and in between the adjacent lacunae, the syncytiotrophoblastic cells persist as trabecules. Now expanding syncytiotrophoblasts, they will erode these maternal capillaries which are present into the maternal endometrium, and the maternal blood will start filling these lacunar spaces, and now it will induce the uteroplacental circulation at day 17 to 22. So that is in the third week our uteroplacental circulation has been established by erosion of the maternal capillaries by encroaching syncytiotrophoblasts. Now, after this uteroplacental circulation, as it is being established, there is one more thing. The maternal blood, it is being delivered to the placenta by the spiral arteries. We know that the vessel was supplied by the straight arteries, and the spongiosum and the compactum layer, they was being supplied by the spiral arteries. So the maternal blood is delivered to the placenta by spiral arteries in the uterus. Now the erosion of these maternal blood vessels will take place to release the blood into the intervillous space. Intervillous space किसने बनाया? The lacunae ने, जो कि डेवलप हो रहे थे प्लेसेंटा में. So erosion of these maternal vessels to release blood into the intervillous space is accomplished by endovascular invasion by cytotrophoblast cells. So the cytotrophoblastic cell layer is there, it will invade the maternal spiral arteries and it will cause erosion of these maternal vessels. Now these cells released from the ends of anchoring villi will invade the terminal ends of spiral arteries where they will replace maternal endothelial cells in the vessel walls, creating hybrid vessels. So the encroaching cytotrophoblast, they will replace the maternal endothelial cells in the maternal capillaries or maternal spiral arteries, creating hybrid vessels which contain both fetal and maternal cells. ठीक है? Excuse me. So to accomplish this process, the cytotrophoblastic cell layer will undergo epithelial to endothelial transition. So the invasion of the spiral arteries by cytotrophoblast cells transforms these vessels from small diameter, high resistance vessels to a larger diameter and low resistance vessels that can provide increased quantities of maternal blood to intervillous space. So that will meet the increasing requirements of the growing fetus. क्योंकि अगर low resistance होगा तो blood flow will be easy. If there will be high resistance, then there will be restricted blood flow. So that is why new hybrid vessels are being formed at the decidua. So there is a condition known as preeclampsia, which is characterized by maternal hypertension, proteinuria, that is protein is being excreted in the urine normally which is not there, and edema, that is uh, uh, swelling of the extremities. So it may begin suddenly any time from about 20th week of gestation to term, and it may result in fetal growth retardation. Why? Because when there will be maternal hypertension, there will be constricted blood vessels, so less amount of blood flow will go in the fetus, so it will lead to fetal growth retardation, fetal death, or death of the mother. So this condition appears to be a trophoblastic disorder related to failed or incomplete differentiation of cytotrophoblast cells, many of which do not undergo their normal epithelial to endothelial transformation. So when there is failure of formation of hybrid vessels, so there will be high resistance blood vessels which will hamper the normal feto-maternal blood circulation. So as a result, invasion of maternal blood vessels by these cells is rudimentary. So there will be less amount of blood flowing to the fetus. Now the development of the placenta, again in continuation, there were columns of syncytial trophoblasts stick trabeculae. So they got penetrated by the cytotrophoblastic cell and they will form the primary villi. So primary villi was forming in the second week of intrauterine life. Then the mesoderm will enter into these primary villi. They will form the secondary villi by the third week. And in the third week, only the tertiary villi will be formed when the mesodermal core will differentiate into the fetal capillaries. So now they are known as tertiary villi. So on the formation of villi, lacunar spaces are termed as intervillous spaces. Now the cytotrophoblast shell will penetrate the syncytial trophoblast and it will form the outer cytotrophoblastic shell. Now, there are some interesting facts that fetal chorion frondosome and maternal decidua basalis, this is fetal chorion frondosome and the maternal decidua basalis together form the placenta, and by the end of third month decidua capsularis, this is capsularis, it will fuse with decidua parietalis due to increased size of the fetus. Then cytotrophoblastic shell, it isolates the syncytiotropoblast from the decidua basalis and helps in anchoring the placenta. So we already know that anchoring villi, they have cytotrophoblastic shell outside it, so it will anchor the maternal endometrium. So what is chorion? Chorion is trophoblast plus extra embryonic mesoderm. We have to remember this. This is chorion. That is extra embryonic somatopleuric mesoderm. And what is decidua? It is stratum compactum of the uterine endometrium during pregnancy. Now the chorionic villi and their branching pattern. So columns of syncytiotrophoblastic trabeculae, they get penetrated by the cytotrophoblast to form primary villi. This is during the second week. Then when the mesoderm, extraembryonic mesoderm enters the core of the primary villi, it will turn into secondary villus. And then when the fetal capillaries, they will appear in this mesodermal core. Now the villi are known as tertiary villi. So the villi towards the decidua basalis proliferate rapidly to form brush-like structure which is known as chorion frondosum, whereas other villi elsewhere it will degenerate. Because यहाँ पे villi क्यों रहेंगे? Because it has to draw the nutrition, and rest of all the places the villi will degenerate and they will disappear, and these villi are known as chorionic leaves. So over here beneath the decidua capsularis, you can say we have chorionic leaf, and at the decidua basalis, we have chorion frondosa. So thus placenta develops from decidua basalis and chorionic frondosum. So ये maternal and fetal part with. So the villi that extend from chorion, that is fetal side of the placenta, to decidua basalis, that is maternal side of the placenta, are called anchoring villi. So anchoring villi will be present only at the chorion frondosum. So several branches arise from the lateral side of these anchoring villi. So these anchoring villi has an outer core of the cytotrophoblast. If you remember, inner was the syncytiotropoblast. Okay. So several branches arise from the lateral side of these anchoring villi that enter the intervillous space. These branches are termed as truncus cori, the main stem villus that connects chorion with the decidua basalis is the truncus cori. Then, like a tree, branches will be given off from it. So that is rami cori. These are the branches of the stem villi, and further finer branches. These are the ramula cori. Then there are free floating or the free villi or the floating villi. These are the numerous villi which floats freely in the intervillous space. So there are four branching pattern you will be seeing. That is the truncus cori, the rami cori, the ramuli cori, and from the ramuli cori we will see free villi or the floating villi. So microvilli of syncytiotrophoblast, which projects into the intervillous space, increases the surface area up to 14 meters square for absorption. So that is the need for this villi. It increases the surface area of absorption. Now there are certain zones of villi as well. This is a tertiary villi. So there are alpha and the beta zones. So the beta zones, the cytotrophoblast layer will separate the syncytiotrophoblasts. So as the placenta matures, slowly and slowly the cytotropoblastic cell layer, it will disappear as in term placenta. So to aid the maternal fetal transfusion or diffusion more correctly. So in the beta zone, cytotrophoblast separates the syncytiotropoblast and the fetal capillaries, whereas in the alpha zone what happens, there is disappearance of the cytotrophoblast and the fetal capillaries lie in close approximation with the syncytial trophoblast. So these zones are known as alpha zones, and these aid the simple diffusion of oxygen and other metabolites at the materno-fetal junction. So exchange of nutrients takes place more rapidly through the alpha zones as compared to the beta zones. Now the what is intervillous space? We are well known with its fact. The intervillous space is the placental space that is formed by fusion of the lacunar spaces of the syncytial trophoblast during second week of intrauterine life. Then these intervillous spaces are partially separated from each other by placental septa, as these septa do not reach the chorionic plate or the fetal surface. So maternal blood enters the intervillous space through the spiral arteries of the deciduoabissalis. The intervillous space is drained by endometrial veins. So volume of one intervillous space is about 140 ml, and so about 500 ml per minute maternal blood is flowing through the intervillous space. So now most important question is most frequently asked question is a placental barrier. So what is a placental barrier? So in the placenta, maternal blood, this one is the maternal side. So maternal blood circulates through the intervillous space, whereas fetal blood circulates through the blood vessels of the chorionic villi in the fetal capillaries. Here, maternal blood is separated from the fetal blood by a placental barrier. So this placental barrier allows the exchange of gases, nutrients, fetal waste products between maternal blood and fetal blood. So it is made up of from fetal side to maternal side, the endothelium of the fetal blood cells. Second is its basement membrane of the endothelium. Then is the syncytial trophoblast, and then comes the maternal blood. So during initial stages of the fetal development, the placental barrier consists of endothelium of the fetal blood cells, basement membrane of the endothelium of the fetal blood vessels, then the mesodermal layer, cytotrophoblast layer, syncytiotrophoblastic layer. That is only the early placenta. But in a term placenta, all these layer, mesodermal layer and cytotrophoblast layer, they disappear, and the alpha zone is there. So fourth month onwards, cytotrophoblast and mesoderm thin out, and thus placental barrier is represented only by a thin layer of syncytiotrophoblastic and fetal capillary endothelium. So we have endothelium, we have basement membrane, and we have syncytiotrophoblastic. Just three layers will form the placental barrier. Now there are certain interesting facts. One is Nita-Book's membrane. You get a one-liner on it. So Nita-Book's membrane, it is all are the modifications of a term placenta. So Nita-Book's membrane is a fibrinoid deposition over the outer cytotrophoblast shell. It makes a demarcation between maternal and fetal tissue. Then there is Rohr's stria or Rohr's fibrinoid stria. It is also a fibrinoid deposition on the intervillous space. It was on the cytotrophoblast shell. It is in the intervillous space of the syncytiotrophoblastic, and it was first described by Wolska. So this layer plays a major role in immunological separation of fetal tissue from the maternal tissue. Why? Because intervillous space is constantly being flushed by the maternal blood. Then is the fetal cotyledons. Like maternal surface has the cotyledons, fetal surface also have a cotyledon. So chorionic plate shows 40 to 60 extensions that extend towards decidua bissalis, and these extensions are called fetal cotyledons. So each fetal cotyledon consists of stem villus and its ramifications. Then there is a Langhans layer. Cytotrophoblast layer is also known as Langhans layer. So some authors also refer the Langhans layer as fibrinoid layer separating intervillous space from the chorion. So all you have to remember all these Nitabugs membrane, the rod stria, fetal cotyledons, and the Langhans layer. So you are most often asked on Nitabugs membrane. Then this is the most important question being asked, that is, what are the different types of placenta or the classification of placenta. So we have classified placenta variously. Firstly, according to its shape, it may be discoid, it is rounded and disc. It may be bidiscoid, that is, there may be two discs. It may be lobed, two or more lobes are there. Then placenta sexensurata, that it shows accessory lobe of the placenta. Then is placenta membranaceae. It is a diffuse and thin chorionic villi projecting around entire blastocyst, not at just one place but entire blastocyst. So that is placenta membranaceae. Then is this one is placenta membranaceae. Then we have circumvallate placenta, that in the circumvallate placenta the peripheral edge of the placenta is covered by a circular fold of decidua. So circumvallate placenta. Then the classification of placenta on the basis of attachment of the umbilical cord. So normally it is attached in the center of the placenta. So when if it is connected to the margin or it is eccentric, then it is known as battledore placenta. If it is attached to the fetal membranes near the peripheral margin of the placenta, then it is known as filamentous placenta. Then, according to the degree of adhesion, that is how the chorionic fundusum has encroached upon. So first is accreta. It is pathologically adhered placenta with the decidua basalis. Normally it is compactum layer, but when decidua basalis is also encroached, then placenta accreta is known. It is known as placenta accreta. Then increta, when it encroaches the myometrium. And finally, percreta is when the entire uterine wall is being encroached. Why these conditions are being very important? Because during delivery, there may be inversion of the whole uterus, may be there. Whole uterus may be come out if we try to pull the placenta, which normally comes out by itself after 30 minutes of the delivery. So that is why these conditions are very important. Knowledge of these conditions are very important. Then a word about maternal serum screening. So there was a search for biochemical markers of fetal status, which led to the development of maternal serum screen test. So there are two tests, mainly the triple test and the quadruple test. So one of its component is alpha fetoprotein, which is alpha fetoprotein is mainly produced by fetal liver, which peaks at about 14 weeks of intrauterine life. So it also leaks into the maternal circulation. So by measuring these levels, we can know about any abnormalities of the fetus. So alpha fetoprotein concentration, it increases in the maternal serum during second trimester, and then its steady decline occurs after 30th week of gestation. So in case of neural tube defects and several other abnormalities like omphalocele, gastroschisis, bladder extrophy, amniotic band syndrome, sacrococcygeal teratoma, intestinal atresia, these alpha fetoprotein levels, it shoots up in the amniotic fluid and maternal serum. So in other instances like Down syndrome, trisomy, there is low level of these alpha fetoproteins. Then these conditions are also associated with lower serum concentrations of human chorionic gonadotropin and unconjugated estriol. So a combination of these three markers are being used for maternal serum screening, that is known as triple test: HCG, unconjugated estriol, and alpha fetoprotein. So alpha fetoprotein will decrease in Down syndrome, whereas it will, in these cases, it will increase. Then in the quadruple test, along with these three agents, there is also a determination of a hormone which is known as inhibin A. So that is quadruple test. So maternal serum screening provides a relatively non-invasive technique for an initial assessment of fetal well-being. Then now what are the functions of the placenta? Firstly is the gaseous exchange, the oxygen and CO2. Gaseous exchange occurs along the placental barrier. Then the transport of the nutrients, excretion of urea, uric acid and creatinine from the fetal blood. Then helps in growth of, get ready for lactation because it secretes human placental lactogen. Then passive immunity, maternal antibodies pass placental barrier by pinocytosis of syncytial trophoblast. So these antibodies provide passive immunity to the fetus against certain diseases, but they are not, the fetus is not protected from chickenpox and whooping cough. Then the placental barrier, it prevents entry of many drugs and bacteria, so it is also having a protective role. Then storage, it stores glycogen, calcium and iron. Then it has various endocrinal function. You get a short note on endocrine function of the placenta. It secretes human chorionic gonadotropin, which will carry on pregnancy. Then it secretes placental estrogen, placental progesterone, placental lactogen. Then human chorionic gonadotropin resembles luteinizing hormone and maintains corpus luteum up to 3 months. Later on, human placental progesterone will take up and then it will maintain the pregnancy. The placental estrogen changes the female reproductive tract to make it suitable for growing fetus. These changes include growing size of uterus, relaxation of various pelvic ligaments, and so on. Human placental progesterone, I told you, helps in growth of decidua and maintenance of the human placenta. Placental lactogen is partially responsible for the uh, development of the mammary gland.